The traditional Indian holistic medicine called Ayurveda has been a universally accepted holistic alternative for over 5,000 years. Despite its long history, rigorously tested and scientifically reproducible formulations of Ayurvedic drugs have remained largely unestablished. We have been researching for over 2 decades the application of green nanotechnology to develop universally acceptable Ayurvedic formulations. Green nanotechnology allows the production and efficient encapsulation of Ayurvedic relevant phytochemicals on gold nanoparticles. The choice of gold nanoparticles is further necessitated by the fact that this metal embodies the iconic medication referred to as Swarna Bhasma, which contains gold particles along with varieties of herbal mixtures. Swarna Bhasma is an ancient Indian medicine with established anti-cancer activity. We have developed an innovative green nanotechnology approach for the production of gold nanoparticles, employing complete scientific control on its formulation for reproducible large-scale production. In this publication, for the first time, we will provide full details on the discovery of gold nanoparticles through green nanotechnology, drug loading concepts, the in vitro anti-tumor properties of this new drug, its preclinical therapeutic efficacy in breast tumor-bearing mice through oral delivery approaches, as well as the first results of pilot human clinical trials in metastatic breast cancer patients. The results include the preclinical in vitro and in vivo investigations in breast tumor-bearing mice, establishing unequivocally that the nano-Ayurvedic drug is highly effective in controlling the growth of breast tumors in a dose-dependent fashion in vivo. These encouraging preclinical results prompted us to seek permission from the Indian government's Holistic Medicine Approval Authority, referred to as Ayush, for conducting human clinical trials in breast tumor-bearing human patients. The primary objective of this pilot clinical trials investigation was to assess if the preclinical results from treating breast tumor-bearing mice can be successfully translated into human breast cancer patients. We report herein the safety profile and assessment of the number of patients who experience adverse events by the National Cancer Institute toxicity criteria at every course. The secondary objective was to determine the efficacy of the Nano-Swarna Bhasma drug, DNA Nanostana as it is referred, in terms of response rate in participating breast cancer patients assessed by radiographic evaluation by response evaluation criteria in solid tumors. Preclinical results of treating breast tumor-bearing mice using the nano-Ayurvedic medicine, which we call it as Nano-Swarna Bhasma, have been successfully translated into treating patients using the Nano-Swarna Bhasma drug capsules along with the standard of care treatment. Patients with the Nano-Swarna Bhasma drug capsules along with the standard of care treatment exhibited 100% clinical benefits when compared to those patients who did not receive this drug. These results, therefore, unequivocally suggest the tremendous clinical benefits of our adjuvant therapy for treating cancer patients.